It started out as institutions were receiving funding in order to enroll individuals into longitudinal studies. And now it's shifted to really investing in ideas, novel ideas, things that can advance the science. And I think it's going to continue having more funding for even more investigators across all stages.
The state has been able to appreciate the progress that's been made. So there's been a great deal of success in bringing federal funding to Texas for Alzheimer's disease. And I think TARCC has been the primary stimulus for bringing that federal funding here.
We have heard about a return on investment of at least 4 to 5-fold, $4 to $5 for every dollar that is put in. But there are additional aspects, and we're desperately short in most of those areas across the U.S.
The existing funds for Alzheimer's and dementia care research is phenomenal, but we can always utilize more. We could use more, we would benefit from more. So many of our efforts are limited just by the available resources given to us. And so with more resources, we could really expand our efforts, expand our reach, and be able to have that greater impact in the community.
Continued investment means progress, and until we find a cure, then we need this investment.
Please welcome Dr. Jared Benge from the University of Texas at Austin.
Well, hello, everybody. Hope you've had a great day today. I know I have. And I should warn you up front, I feel like my talks should come with a disclaimer. Typically, even at neuropsychology conferences, the measurement people get relegated to Saturday morning 7:30 a.m. slots because of the risk of falling asleep during measurement talks. And so, and you could think I'm joking, but you can ask Robin Hillsbeck. We're the breakfast club at conferences. So as we talk about measurement things today, be warned, you can also do your own kind of multiple trial to see, you can watch the YouTube video later and see if I'm more effective than Ambien for sleep. So I recommend you try that. But what I'm going to talk about today is something that I'm very passionate about, and it's measuring daily activities. And how do we measure this in those impacted by ADRD now, but where do we need to be going, and how is TARCC supporting that work? So I have no conflicts of interest. You can see my other funding currently. I will talk about the results of some of these other studies that are ongoing here. So we're going to talk about, I know we have a room full of individuals that go all the way from basic science to social researchers and everywhere in between. So I'm going to talk about a concept that we may not all be on the same page about, but the idea of instrumental activities of daily living. How do we measure what real people are doing in the real world? And we're going to review some of the limitations of existing measures, and then talk about our newly funded TARCC collaborative grant about how we're going to be addressing these questions. I will tell you that our funding started January 1st, and it is January 23rd. So I don't have the final results to show you, but I'll show you where we've been heading and hopefully get you as excited about these questions as what our group is. But before we do that, it is TARCC, and we should talk about what an amazing place Texas is to do this work. And as I was putting together these slides, what really struck me was how the investigators, the clinicians, the scientists all across our part of the state have influenced the work that we've done and these ideas. And as I was going through and I was preparing these slides, we're talking about ideas that I've come up with in collaboration with my dear friend and colleague at Baylor University, Mike Scullin, data that we've collected in collaboration with the Georgetown Neuroscience Foundation north of Austin, led by Mike Douglas. We have this amazing team that put up with me and all of my randomness here at UT Austin. We've got the individuals at UT San Antonio I'll talk about next. We'll even give a shout out to my friends at University of Houston, Luis Medina and Stephen Paul Woods, who have been instrumental in shaping some of these projects. And so this I-35 and I-10 corridor is the reason that these ideas are in Texas and being developed in Texas and will be brought to Texas. But most importantly here, I'm going to talk about the collaborative research project that we're doing that kind of is led here at UT Austin in collaboration with Alyssa Aguirre, who you heard earlier, as well as Ceci Zuniga, who's there and keeps me out of trouble. Thank you, Ceci. As well as our colleagues in San Antonio, Dr. Robin Hilzebeck, Dr. Amy Werner, and Dr. Gabriel Romas, who are helping with this project and really bringing a collaborative approach to these projects here. So in the day and age of all of the excitements of biomarkers and all these kind of basic science approaches and digital technologies, why on earth am I going to stand up here and talk about questionnaires and questionnaire development? All right. So I think to bring this to bear for everyone in the room, whether you're a basic scientist all the way up to a kind of a systems or sociologist, why look at these activities of daily living and their measurements? I want to go back to my first experience of falling in love with science. All right. So I think most of y'all probably have that moment where you realized you were falling in love with science or you were going to be a scientist. Another way of framing that is the moment you realized you were a nerd. So for me, I was 7. My grandparents took me to the big city of Oklahoma City, to the Omniplex, one of those obligatory children museums. All right, I'm seeing some nods in the congregation, so I think a few of us have been there. Okay, and what was there was a video that was played by a guy named Eames. They make chairs and office furniture. So I don't know how they ended up in a children's museum, but there you go. It's a video called The Powers of 10. I recommend that you go check it out on YouTube. But The Powers of 10 is this fascinating video that starts with the picture in the middle of a couple that is having a picnic in the park and zooms out further and further at powers of 10 and zooms in at powers of 10 to the same scene. So you see the couple in the park all the way out to where they're at in, I think, Illinois, where they're at in the globe. As you keep moving out powers of 10, you see where we fit into the whole universe. And then when you move in at powers of 10 all the way down to the individual hand, the individual cell in the hand, the individual cell, the individual nucleus, and all the way down to subatomic particles. And I fell in love with that video. Again, that's probably a poor prognostic indicator for my social skills development. But I sat there and what that meant to me then and what I still think about to this day is that every phenomena can be understood at very different levels of understanding depending on how far you zoom in and zoom out. Okay? So when we think about Alzheimer's disease here means different things to your research groups or to your own personal lives, depending upon how far you're zooming in and zooming out. And so we've moved toward this biological definition of Alzheimer's disease. So Alzheimer's is these, amyloid plaques and neurofibrillary tangles, right? Well, okay, but those things impact how cells are functioning, all right? Those cellular dysfunctions impact how brain networks and structures work. Those in turn impact cognition. Cognitive decline impacts how people function day to day. That has an impact on family and carers. And that then leads to changes in our society and our economy, right? All of those levels of analysis of Alzheimer's disease are true. And they all matter, right? You can imagine these microscopic pebbles getting thrown into the pond that is humanity. And what you see as the impact of Alzheimer's disease depends upon how far out you zoom and what waves you look at here. Okay, so when we talk about Alzheimer's disease and the exciting kind of advances in the biological definition of Alzheimer's disease, this has been the excitement of the last 2 years. Okay, but if you'll notice, there has been a lot of trepidation with that excitement, because how much is this helping becomes the question that people keep asking. Patients ask us, the research fields ask each other, and this gets at the idea of how do functional changes get measured. This is a measurement issue here, right? And when you work with individuals who have ADRD or their families, right, what are their actual goals, right? As a neuropsychologist, I've never had a patient come up to me and say, I would like to improve my clock drawing, right? That has yet to happen, all right? And you know, as you think about changes and the excitement about changes in these biomarkers and these sorts of things, these are all horribly exciting. But what do patients and families actually want? Right? And so the shared goals of individuals who are impacted by ADRD are to function well, independently, and to not be a burden on their families. Right? This is the core of what we do. If we want to make an impact day to day, we have to take these goals in mind for those that we serve. Right? So this is why I'm passionate about this topic, is that if you want to treat the things that matter, if you want to measure the things that matter, you have to be able to keep function at the core of what we do. Okay, so how are we currently measuring IADLs, right? So my very first grant when I was a intern, psychology intern, I was taking pot shots at the CDR, the Clinical Dementia Rating Scale. It's good to see from some of the posters that other people are still taking pot shots at the CDR. It is a long tradition, a rite of passage for neuropsychologists there. We have a lot of tools. As a field, we've been very conservative about advancing our tools. So there's a handful of tools that we use. CDR tends to be a good co-endpoint for a lot of clinical trials. And then we have a series of questionnaires that get used as well. One of these is the Lawton-Brody ADL scale. The Lawton-Brody is from 1969, and it's one of the most commonly used ADL measures. I've used it for years in clinic and studies. It's the core ADL measure in the TARCC database here. If you want a good read, if you want an interesting read, go pull the original 1969 Lawton-Brody article. I don't think any of you are actually going to do that, but I did because, again, I'm the kid in the science museum. So I have a new life goal. Is to write a paper, a peer-reviewed paper that has as honest a paragraph in the first here as what Lawton-Brody says. The present state of the trade seems to be in which each one creates a scale for their own preferences and says none of the rest of them can work for us. Yeah, that's true. And they did it too, all right, but their idea won, all right. So their idea has had over 23,000 citations, which is exactly 21,000 more citations than my entire publishing career has had. So I want to be careful casting stones here, all right. But this scale was made in 1969, which is a different era. And I'm going to start planting these seeds and then systematically try to water them and help them grow here. This scale came from a time where there were very strong assumptions about gender roles and activities of daily living. Guys, we don't come out looking good on the Lawton-Brody because, quote, these are not applicable to us for things like cooking, cleaning, those sorts of things. That's a very strong assumption, which is not held up over time, okay? The demographics of this sample weren't even described. You can't find a demographics table about in this paper about this article that has become the lingua franca of activities of daily living over time. That being said, it is everywhere in the TARCC database and many other huge databases, right? So one of the questions, if you're going to wade into the space, is not just make a new questionnaire then. It's how do you build something that can not throw the empirical baby out with the bathwater? That's an interesting question for me anyway, and hopefully for y'all. Okay, so let's talk about a few of the items on the ADL just to illustrate some potential problems here. And like the good neuropsychologist I am, let's do some IRB non-approved convenient sampling research of our own here to look at this here. So one of the items asks about ability to use the telephone. And so the exact description that you would give to a caregiver is, can this person operate a telephone on their own initiative, look up and dial numbers, etc. Okay, so let's look at this one. In the past week, raise your hand if you've actually typed a number into your cell phone to call someone. All right. I'm going to say well less than half. All right, let's ask another version. How many of y'all would not answer a call if it's not in your contact list? Yeah. All right. This is an idea. We can even talk more about the idea of looking up. How many people? This is scary because I'm old. How many people have never actually looked up a number in a phone book? All right, that's fewer than I expected, but still more than makes me feel comfortable. All right, so you can see that one item. Now that's funny. Okay, it's changing. Imagine if you're filling this out about your grandparents, right? How do you answer that question, knowing the fact that, how does this actually fit into how they're using their smartphone here, right? Here's another one: ability to handle finances, manage financial matters independently, budgets, writes checks, pays rent, bills, goes to bank. Okay. How many of y'all have been to a bank in the past month? All right. That is well less than a quarter. All right. How many of y'all have written a paper check in the last month? All right, Houston, we're starting to see a little bit of a problem here, right? You can see that this shift is starting to change. This goes on and on. You think about modes of transportation. We struggle in clinic with this a lot. We routinely see people who they're still driving, but they have a car that has all the even my 2019 Corolla has lane departure warning, and it's very basic. Neuropsychologists don't get the upgrades, right? So lane departure warnings, GPS systems for navigating familiar places. Is someone who's using those things independent, not independent for transportation? Can you tell, all right? Shopping, right? Which one's more cognitively demanding, do you think? To go and shop online, to use an app to get groceries delivered, or to actually physically go to the store, find the items on the shelf, check out with them, right? What does it mean to be independent for shopping these days, right? So hopefully you can start to feel a bit of that tension about we might be having some problems here. So this is, when you look at kind of the ADL assessment space, this is my attempt to have a snarky first paragraph in a review paper. But you look at the overall timeline of when our ADL measures were developed. So Lawton and Brody in 1969, FAQ, which is the core ADL measure for the NAC database, was made in 1982. I was made in 1980, and I'm looking pretty long in the tooth, right? So you look at kind of these old kind of measures that have been out there for a while versus the rate of change that has happened in activities of daily living. So you see the personal computer really coming online in the 80s. You start to see the first iPhone coming out in 2007. You see by 2015, Bank of America saying 2/3 of all their bills are being paid on auto pay. Right? If someone has all their bills on auto pay, are they independent for financial management? Are they not? Can you tell? Right? So there's a little bit of disconnect between when our measures were developed and the rate of change and how we're actually performing a lot of these daily activities. All right. So when you look at data from our friends up in Georgetown, north of here, in a suburb here that has a community called Sun City that is predominantly older adults, we did a simple measure where we looked to see kind of how are individuals managing daily tasks. Are they predominantly using digital approaches? Are they predominantly using analog approaches, paper-based versions, or both? Right, and what you'll see is me skipping slides dramatically and then pushing the correct button. But what you'll see is that for some tasks like managing appointments, managing to-do lists, you'll see individuals that still frequently use kind of a hybrid approach where they'll use paper and digital techniques. The mean age of the sample was like 74, I believe. But you can see that a number of activities of daily living, right? So things like navigation, paper maps versus GPS, definitely skewing toward more GPS use, paying bills, definitely skewing toward auto pay, online pay, making purchases, online shopping is kind of increasing in frequency. So you can see that this is not just a theoretical idea. We're actually starting to see these play out in older adults. Okay. This trend seems likely to get stronger over time. This is a hard slide to read, but this is some data from Houston that the amazing Dr. Miss has got under revision right now. But what I'll draw your eye to is the colors, right? So we have a younger adult sample and an older adult sample, right? And yellow is a hybrid approach. Individuals saying that they take a digital and analog approach for your daily activities, right? Blue is a pure digital approach, the percent of the sample using a pure digital approach for their daily activities, right? And green is a pure analog. I don't use anything digital for this task, right? And what you'll notice, right, is a whole lot of blue and yellow in our older adult sample and in our younger adult sample. And you can see that green is pretty much disappearing for younger adults, meaning that whatever we're getting by with now probably won't last into the next generation of people that are going to age into the range where neurodegenerative diseases start. Okay, so now some individuals might say, well, that's great for other people, but digital technologies are just too hard for people that we serve in clinic, people with MCI and mild dementia to actually use. And so if you're one of those people, you were likely reviewer 2 back in 2016. So it's always reviewer 2. I don't know about y'all. I feel guilty whenever I end up being reviewer 2 because I have an ongoing fight with the anonymous reviewer 2 of every article and grant I've submitted. But the way back in 2016, we submitted our first R21 looking at smartphones as cognitive prosthetics. That's another angle of our work, is using smartphone apps to help with prospective memory. And the rejection came with this little nugget here. Many individuals may prefer more traditional methods to achieve the same goals, especially in those who are older or with conditions to be more easily intimidated by today's technologies, including smartphones, leading to poor compliance and adaptability in using technology. So I got mad. And as a researcher, what do you do when you get mad? You write a paper. So let's go get data to do this rebuttal here. So way back, we published this in 2020. The data would have been collected around 2017. It would have been collected probably a day and a half after I got that review back. But what we saw back then when we went to our clinical samples of people with MCI and dementia, we saw that over a third of individuals were using, with these diagnoses, were using their smartphones regularly for cognitive and general activity tests. We updated this with some support from the Alzheimer's Association more recently in a paper that just came out. We saw that number has gone from about a third of our patients up to applicability of digital daily activities for people in clinic climbing above 80%. Right. So it's no longer the case that just because you're older and have cognitive impairment, these digital technologies are just too much for you and you're not going to use them. It may be harder, right? It may present new challenges, but to say that it's not a part of the lives of those that we serve is dramatically misrepresenting the lived experience of people with MCI and early Alzheimer's. So obviously, technology changes how we do day-to-day activities, right? But let's also think about the demographic and social trends that have changed since 1969, right? So I think all of us are aware of the importance of bringing everyone into ADRD research, right? So that factors such as ethno-racial backgrounds, gender, educational status, rural versus urban status, linguistic status should not be a barrier to participation in research and clinical trials, but that also means we need instrumentation that captures these day-to-day activities better. And so one of the co-investigators on the grant here, David Gonzalez, he is a good friend of mine, and we actually started thinking about this. In the NAC database, we went to look for those items that were rated as not applicable, never did, right? A lot of papers have been written from the ADL database about how these ADL measures work, right? But what you don't see is individuals who aren't captured in the data, right? Where a particular item was not applicable for that person. And so we tried to model not applicability for the different items here, right? And we demonstrated this intersection effect, but where a lot of different demographic identities can predict these specific ADL items being not applicable, right? So let's go back to the cooking example. There's still a gender bias in the cooking item, right? But what you see is this kind of intriguing interaction effect where women who have higher education were more likely to have the cooking item not apply to them because they're out doing their careers and those things, right? Versus it was the opposite in men, right? Where men who had more educational status were less likely to have cooking items not applicable to them. You saw all sorts of fascinating intersections where these different activities of daily living items were not applicable at the intersection of these increasingly diverse cohorts. Okay. With that, we also run the risk of bias with some of these items. So in other words, if you imagine that each item on an activity to daily living scale gives you some degree of information about an underlying construct of activities of daily living limitations, you can model through something called DIF, differential item functioning. You can actually model and measure how the items might give you a biased indication of that underlying ability. And we saw a small but measurable amount of diff for a number of different items, virtually all the items on the FAQ, right? But we also saw kind of interesting interaction effects, again, between different ethno-racial group, language, age, education, gender factors in driving this diff. So again, these items are performing differently depending on your background. Okay, so hopefully you're sitting, I'm going to sound like a psychologist. I always apologize when that happens. I hope you're sitting with a little bit of that tension there about some of these things, right? On the one hand, focusing on functioning and IADL measurement should be the core of what we're doing. We want real people to function better in the real world, to be independent and do the things that they want to do. This is what we want, okay? But our existing measures were created a long time ago, before things like technology and representativeness and these sorts of things were well understood. We also know that these scales sit at the core of these big databases that we look at, and we don't want to get rid of data that has been used for years, right? We don't want to throw the baby out with the bathwater here. I hope you're also sitting with a little bit of attention that this is not going to be a one and done type of problem. In other words, it's not like technology is going to stop developing today and that's it, right? Like, there's a very real chance, as much as I love research with smartphones and those things, that I end my career and nobody has a smartphone. I think that's a possibility. I love road trips. I am really good at road trips. I may not always be driving because my car does it for me, right? So I think this pace of technological development is going to continue to change. So even if we fix the problem today, we just end up in the same problem tomorrow or a year from now or 10 years from now. So I also want to point out that what we're asking from activity of daily living measures is a lot different now than what it used to be. So with the advent of these biomarkers, we've talked a lot today about prevention. All right. So with prevention means that you're hopefully going to start in research trials and then in clinical practice, treating people before they're at the dementia or even the MCI level. The new diagnostic staging criteria that came out included this pithy little thing called transitional cognitive decline, which is something between normal and MCI, right? So with that, you should have no or minimal functional impairment versus some mild impairment, but still independent versus dependent, right? As a neuropsychologist, those words cause me anxiety because I don't know what those words mean or how to measure them. So we're going to be asking these questions about subtler and subtler changes and impacts earlier in the disease course as the field moves forward here. So, and again, I'm not putting all my eggs in this basket, right? Y'all heard Dr. Miss talk about the Texans app. That's one of the RO1s that we're working on. I would love to just directly measure what people are doing in the real world. And I think that is an approach to everyone being their own baseline is a future approach for this. But there's always going to be a need for something that's easy to deploy, easy to manage, easy to get out there, and easy to measure that also links with existing databases to make our data talk across generations of the ADRD disease space. Okay, so hopefully you feel vaguely angsty. I only see 4 people asleep, which is pretty good for one of these measurement talks. All right. So how are we going to set about actually addressing these problems, right? What are some ways that TARCC is helping us push this field forward? Okay, so there's 3 core aims to our collaborative research grant here, right? So the first one is we're going to leverage the data we already have. The amazing gift that is the TARCC database and the richness of that database allows us to ask some interesting questions about how the existing Lawton and Brody items are working and which ones in particular we need to revise, right? Second, we're going to talk to the people that we're actually asking to make these ratings to figure out how to make them better. As y'all probably have learned by now, me being quiet is not something that comes naturally to me. But for this sort of work, we want to talk to the real experts, which are the people that are going to be making these ratings, which is caregivers of those living with cognitive impairment. And then armed with an understanding of how items are currently working, what items we're missing, we're then going to create new items, bring them into clinic, and link and equate with existing measures. In other words, we want to use this as an opportunity to show how we can build and modernize instrumentation to not throw babies out with bathwater, but to create a living, evolving instrument that changes as the needs of day-to-day activities change. So let's drill down into this a little bit better. Okay. This is where I tend to lose people, and so I promised I wouldn't say theta or discrimination parameters or parameterization in general. People don't like it when I say those things. But if you've taken the MCAT, the SAT, the GRE in the past like 20 some odd years, you have already experienced something called item response theory. Okay, so in item response theory, it is this idea that you don't create scales. There is no one SAT that everybody takes. Instead, each item has a particular difficulty item. So like if you're trying to create a measure of mathematics, an easy item would be 2 + 2, right? If somebody gets that right, they have at least this amount of ability in mathematics, right? And if you ask them to take do a differential equation, right, you at least have this amount of mathematical ability if you get that item right. And so people on the MCAT, people on SATs, they don't all get the same measure. They get a set of items that each item predicts your ability level, and you get more and more items until you can precisely estimate an underlying ability level. And so by using the existing TARCC database, which is large enough and a very diverse sample, allows us to get these IRT parameters established for this measure. This will become important for AIM-3, but this also allows us to check differential item functioning and these other sorts of parameters that we can look at to see if there are particularly problematic items on the existing Lawton and Brody. Okay. Then, armed with this data, knowing we've got some problematic items, we also do this kind of mixed methods approach where we're going to go out and we're going to interview real-world caregivers. And so we're going to do a think out loud approach, which is a qualitative approach where we have people take the existing Lawton and Brody about the person that they care for and actually think out loud, particularly thinking about clarity, applicability of items. Tell me what you mean. You said that was weird. Why was that weird for you? Why doesn't this fit with the one that you love, right? We're going to ask specifically about how technology is used to perform activities within these domains. We're going to ask about family and cultural and linguistic norms and how they influence how these activities of daily living are performed. In addition, we're going to ask what I think is oftentimes the most important question if you're working with human subjects, which is, what did I not ask about that I should have? That always yields the most interesting data when you're doing anything qualitatively here. And we're also going to ask caregivers, who are the ones that frequently fill out these ADL scales, how they would place different labels of severity on this new ADRD scale continuum, this 0 to 6 continuum, where we're looking at making these distinctions between none or minimal functional impairment versus mild but still independent versus not. Like, what labels would you want a caregiver to be using to actually answer that question, to give that sort of rating? So we're going to leave the qualitative stage with a rich data set, which we'll need to combine to kind of identify how to refine existing items. And we're armed with our statistical knowledge of how the existing scale is working, right? And then that leads us to AIM-3, which is this idea of bringing this information back into the clinic, right? So with an IRT approach, what we can do is we can actually see how our existing items correspond to the items that are novel, that are created. We can start demonstrating how we can link and equate these things together. We can start looking at sensitivity and specificity of the revised item sets to the presence of functional impairment and change over time. We start bringing this into clinic. We also start seeing how we can stitch these different data sets together with these different sorts of ADLs. And so this is going to be important as we pursue kind of future funding to modernize this space. It's the ability to work together across institutions, but also to show that it's feasible, that it's possible, and that it's good to actually evolve these measures over time. Okay. Let's see here. So our ultimate goal here is to measure what matters. We want to measure what matters most. We want to measure it well. The last couple of years have been this incredible breakneck pace of change, and we can measure aspects of Alzheimer's disease in a manner that, in a way I couldn't have dreamed about when I entered into this field. With this new biomarkers and biological data, we have this fundamental unlocking of how brains are working at this molecular level. That's amazing. I want to bring that same level of rigor to the measurement of what matters most. And we're going to have to do this as a field in order to be able to meet the goal of understanding the earliest and the subtlest changes of these diseases, and whether our new treatments are working to keep people as independent as possible. And ultimately, we're doing this with the state of Texas, with the talent and ability and the incredible engagement from those that we serve that Texas brings. And so this is a Texas born and bred project. And I appreciate TARCC, and I appreciate all y'all for the ability to get this done. Thank y'all.
Please welcome Dr. Agenor Limon from the University of Texas Medical Branch at Galveston.
Thank you very much, everybody, for being here. Thank you, everybody that is watching us online. And thank you for the organizers to give me the opportunity to talk about the work that we are doing in the Mitchell Center and the Moody Brain Health Institute that we are going to be talking about at the end of the talk. About this, it's working. Yeah. So I don't have to go into the details here for what we are going to be talking about, the pathology of Alzheimer's disease, but I just want briefly to mention the characteristics that we can observe in Alzheimer's disease. We know that there is cortical shrinkage, neurodegeneration. You can see in the images to the left panel how we lose 1/3 of the mass of the brain. So that will indicate that we are going to have problems in many brain areas that are affected in that disorders. We also have macroscopic plaques that you can see in the right panel next to the neurofibrillary tangles that are known by hyperphosphorylated tau. So we have then the classic N+, A+, T+, and we know about the stages of the neuropathological changes that we can observe as Thal, as CDAT, and BRAC for this pathology. We also know that one of the most important correlates of neurodegeneration and cognitive impairment is going to be neurodegeneration. And we also have now information that the synapses are going to be an important factor in the presentation of those symptoms. We also know that there is a large body of evidence that late extensive neural death is linked to the toxic effects of oligomers, of tau oligomers, of A-beta oligomers. So what are those? So there is a lot of evidence that has been showing that proteins like A-beta and tau start to aggregate. Why they do aggregate? Because for reasons that chemical structure people knows better than I can ever know, but they start to aggregate. And they have dimers and soluble oligomers, protofibrils, fibrils, and then they form the deposits. We know that the soluble oligomers, by work made by several groups, most important, some of those by Dr. Cayet's laboratory Cayet's group when he was working with amyloid, now with tau, they produce changes in the membrane that change calcium homeostasis, that bind to many proteins that alter the function of many processes in the neurons and many other cells. But particularly, this is some of the early experiments shown by Dr. Tegratella, that was the previous director of the Mitchell Center, and now is the director of the Moody Brain Health Institute. He started to apply, for example, in cells in culture. You can see in blue the hippocampal neurons in culture, and if you apply labeled amyloid oligomers, you can see that those little stars that you see on the neurons, those are spines. And so the A-beta oligomers are binding to the spines that are mostly excitatory synapses. So then all the oligomers that are being around the plaques and the neurofibrillary tangles are binding to the synapses, but also are changing the function. To the right, you can see a typical experiment when you take a brain of the hippocampus, you slice it, you cut it, you put some electrodes, and you can start recording the activity of the synapses. After high-frequency simulation or a protocol that simulates when we are having a memory, then you have an increase of this activity that is called potentiation, of long-term potentiation. That is a proxy of synaptic plasticity. If you start to apply A-beta oligomers at different concentrations, as you can see in the plot, the function of the plasticity is dramatically reduced. So then A-beta oligomers are important because they bind to the synapses, they disrupt the function, and they disrupt neuronal communication. If we don't have enough activity or communication between neurons, all the functions that are associated to those brain regions are going to be disrupted. And because all the behavior that we have for daily function depend on the neuronal activity of the neurons, then those functions are going to be affected. But we know that we know also that mutations in presenilin 1 and mutations of the APP are going to produce all the phenomena that is associated with familial Alzheimer's disease. But familial Alzheimer's disease is just a fraction of all the cases of Alzheimer's disease that we can observe. The majority of them are very complex. We have factors that are very important, for example, vascular insufficiency, metabolic syndrome, obesity, heavy metals, mitochondrial impairments. Some of those, we don't know exactly how they are affecting the brain. We know they are factor risk. We have a lot of evidence now, and some of those are actually modifiable risk. So we have seen that 45% in the latest report are risks that can be modifiable. But there are others that we cannot do anything so far. For example, old age, or ApoE4, or some infections can produce neuroinflammation. So those things produce a very complex problem to be treated pharmacologically and addressed in Alzheimer's disease. So that's the challenge. So in 2012, Dr. Telatela, together with other groups, but in particular, Dr. Telatela, and I quote, wrote that certain individuals referred as non-demented with Alzheimer's disease neuropathology, NDAN, remain cognitively intact despite the presence of plaques and tangles that would normally be consistent with fully symptomatic Alzheimer's disease, A+, T+, N+, according to the most recent NIA-AA classification. And end of the quote. So these early descriptions of individuals that have severe neuropathology of A-beta and tau and are almost indistinguishable from those that have Alzheimer's disease, but they remain cognitively normal. So that is a very, very important observation. And here in the left panel, you can see histology of the frontal cortex of controls, Alzheimer's disease, and NDAN. And in green, you can see A-beta plaques, AD, and NDAN. You can see they are very similar in size and abundance. But the cognitive impairment in one or the cognitive score of the mini-mental status examination is 1, and in the other is 28. That is in the normal range. And in below, you can observe phosphorylated tau that is also very similar between the Alzheimer's disease and the resilient individuals. If you take a histological slice of the frontal cortex, you can see also you can see plaques and neurofibrillary tangles. And in the right, you can see just very briefly the demographics indicating that the only difference there is that they don't have cognitive impairment in the resilience. So if this is the case, it's very, very important to understand what are the mechanisms that these individuals have that despite having severe abundance of pathology, they still remain cognitively intact. Because if we can understand how those mechanisms work, then we probably can we can find targets that we can use to increase resilience in people that are vulnerable to Alzheimer's disease. And we know that this strategy could work because there are people that are actually resilient. In the Mitchell Center, Dr. Tagliatella, in collaboration with other researchers, have been publishing many of the mechanisms of these individuals. There are changes in neurogenesis, there are changes in microglia, there are changes in many processes. And what we have learned in these studies is that synapses that are usually binding A beta oligomers in Alzheimer's disease, in the case of resilient individuals, they are not binding A beta oligomers, same as the others. And the same happens for tau. If you see in the left panel, there is a Western blot where you can see the oligomers for tau are very clear in Alzheimer's disease, but in resilient individuals, they are absent, similar to the controls. And in the right, there is some unpublished data using the parietal cortex, where we are measuring tau and by mass spectrometry. Same thing, high severe pathology, but only the tau protein is observed high in Alzheimer's disease, not in control, not in resilient. So this suggests that the synapses in resilient individuals may be not accepting the tau oligomers, or maybe they are processing very fast, or maybe they are being removed. It may be possible that also some of these oligomers may have the capacity to aggregate, but they are not toxic. So there are several possibilities. Which one may be working here? So we don't know. We have evidence of some of these processes, and I'm going to be talking more in the next slides. But the question then is, what is happening with these synapses? Because it's very evident that the tau oligomers are not present at the moment when we are doing the experiments. So then I'm taking this diagram from Dr. Edwards, where one of the hypotheses is that you have thousands of synapses in a single neuron. And then if you are losing some synapses, we have compensatory mechanisms that compensate by homeostasis the function of others. Those homeostatic mechanisms are very, very important, and a neuron has very redundant mechanisms to protect the neuron as a whole. So in this hypothesis, it is possible that the concentration of A beta oligomers are very, very high around the plaques. So if that is the case, and what we know in histology analysis is that the synapses that are close to the plaques are dystrophic, are being lost. So then the question is, okay, concentrations of oligomers near to the plaques are very, very high. So those synapses are going to be affected and is going to have calcium-induced homeostasis. The neuron has now to decide, well, they don't decide, but has to produce some mechanisms to avoid the neuronal death completely. So one possibility is to shut down those synapses. But the most important thing is that there are microglia that can go to the synapses and remove them. And by removing, is taking away the calcium-induced homeostasis that is happening in that and is protecting the whole neuron. If that is the case, then we should be able to see it. And Dr. Fracassi, that is in the Mitchell Center, has worked very hard in collaboration with Dr. Tagliatella. And what they have shown is that in Alzheimer's disease, we have alterations of microglia. However, in resilient individuals, microglia is more hyperactive. In the left column, you can see the microglia, and in the right column, you can see the microglia in Alzheimer's disease, and in the right side, you can see the microglia in resilient individuals. And as you can see, there are some changes in the green part. Next, in the bottom panel, you have CD68, that is an enzyme that is in lysosomes of the microglia that is hyperactive. And in magenta, you have plaques. When you merge them all, you can see the changes that are happening. And in NDAN, so you have larger abundance of microglia that are hyperactive, as you can see in the plots, but also changes in the form and the structure of these microglia. So right now, she's doing a collaboration with the Wyss Center in Geneva to understand by expansion microscopy and by artificial intelligence, what are the changes that are happening in these microglia, and whether they are different compared to the resilience, compared to the individuals that have Alzheimer's disease. Another thing that is happening in resilient individuals is that the structure of the axon and the dendrites are very well conserved. by using MAP2 or beta-tubulin that are markers of axons or dendrites, then you can see in the plots that are already changes in resilient individuals that are higher than in control. And you can see MAP2 in the left panel. So this indicates that there are some changes in resilience that are compensatory, going beyond the controls. And in the beta-tubulin, there are no changes with the controls, indicating a maintenance of these processes. Another experiment that is important for this is that the damaged synapses are phagocyted by hyperactive microglia in these resilient individuals. So one way to understand that is that microglia is pruning synapses that are excitatory, and you can measure that by using PSD95 markers, antibodies versus PSD95. PSD95 is an anchoring protein that puts glutamate receptors in the synapses and maintains those AMPA receptors and NMDA receptors. So these experiments were showing that if you measured CD68 that is in the lysosomes of the microglia that is hyperactive, you will see co-localization with PSD95. And in this, basically, you can see in yellow, the co-localization, indicating that PSD95 is inside these lysosomals that are in the microglia. So that putting all these together indicates that microglia in resilience are more hyperactive beyond the controls, are actually eating synapses that seems to be dysregulated, and is keeping the neurons from dying and preventing very early on. Now, one of the questions, though, is microglia is selectively pruning synapses, but how does they do that? So there are some proteins that are put in the synapses that are signals for the microglia to be removed. However, microglia are very, very sensitive. They can actually target excitatory synapses or inhibitory synapses depending on the environment. So they are very, very, very well they are very well enriched with receptors, synaptic receptors, GABAergic receptors, glutamatergic receptors, signaling that can detect the levels of neurotransmitters of function around them. And they can actually prune those very specifically. So the question now goes to what is the functional status of synapses in resilient individuals? And there are experiments being done in animal models that are very, very, very important, because resilience is an important characteristic that you can see in many species. But in the particular case of the humans, where we have a large variety of factors, how is the status of this functional? How is the functional status of these synapses? It's complicated so far to have an animal model based on that. But it's going to be important when we have data that can use the animal model and then validate. So I think they are very complementary to each other. So that's why one of the approaches that we use in the laboratory is the microtransplantation of synaptic membranes. So this approach it was done precisely to detect and measure synaptic receptors that were in the human brain, and they were working and we can transplant them. The way it works is basically you have a postmortem tissue brain that has been frozen for many years, and then you can take the brain region that you were interested, hippocampus, parietal cortex, frontal cortex, take a piece of it, isolate synaptosomes, homogenize, and then transplant in some cells. We use frog cells because those cells are very, very large. And once you inject the synaptosomes, you can actually break them up to create proteoliposomes, and they fuse with the membrane of the oocyte. The advantage of this method is that those receptors are embedded on the lipids that they were in the human brain. They have any A-beta oligomer or fibrils. They have changes already in the lipids. They have anything that was happening in the environment at the moment of death is going to be there. Epigenetic changes, translational changes. So that's one of the advantages. And then we transplant those into the oocyte, and then by 2-electrode voltage clamp, we put just 2 electrodes, apply the drugs like GABA, glutamate, we can record the function of these receptors. And this is how they look like. In the left, you can see a preparation of synapses by electron microscopy. Then we have enrichment. And when we break them up and inject them, these are the 4 types of major responses that we can observe. We can observe responses to GABA, that is the most abundant inhibitory receptor in the brain, GABA A. We can record AMPA receptors that are the most abundant excitatory. We can record NMDA receptors that are extremely important for synaptic plasticity and a major target of drugs, and voltage-gated calcium channels. So we have those that can be recorded if we put a lot of effort at the same time in a cell. Usually, we can record GABA and AMPA very easily. And GABA and AMPA are very important because they actually are fast receptors that manage the excitation to inhibition balance that has been shown to be affected in not only in Alzheimer's disease, but also in other disorders. The advantage of this is that we can activate the currents. You can see that GABA currents are the most abundant in the brain. They have larger responses. You can see in these traces, GABA receptors are like 20, AMPA receptors are 10, it's a fraction of that, and NMDA receptors is a fraction of the fraction. They are very small. So this is observed in all the human brains that we have recorded, depending on in cortical region. So this is one of the advantages that we can have immediately. And then you can see the biophysical characteristics and also do pharmacological experiments. Here, if you apply ketamine to the same onset that has all the receptors as we did in this experiment, you see that ketamine doesn't do anything to GABA, doesn't do anything to AMPA responses, and is an antagonist of NMDA receptors. Now, when we published this for the first time, we have a positive response, but it was interesting because we tried. And I'm saying this tried, very clear, that to publish this for the first time, but it was, the people didn't believe us at the time that we were able to do this type of experiments. However, when we were able to do it, we have used this technique with very success, and we have shown, for example, that in humans, there is a dramatic reduction of GABA receptors in Alzheimer's disease. At the time when we published these experiments, GABA receptors were considered to be well protected in Alzheimer's disease, and that probably was associated with the binding to TSPO, that is inflammatory, and is peripheral benzodiazepine in experiments that were using binding as a metric of abundance. But when we were measuring the function of the GABA receptors, we saw a large reduction of them. The other thing that we observed, that we published, is that there is a loss of synapses in Alzheimer's disease, but the synapses that are being lost are excitatory and also inhibitory ones. And the inhibitory ones, the loss of inhibitory ones is larger than that of the excitatory. So they produce an imbalance in regions that are very important for the whole function of the brain, like as the default mode network. So we observed also that this level of the imbalance correlates with cognitive function in individuals. The more the imbalance toward excitation in regions like the parietal cortex, the worse the cognitive score. Same thing in the temporal cortex. But however, that's not the only case. In other brain regions where we observe reduction of activity, like hypometabolism in the frontal cortex, we observe a hypofunction of these receptors, of this imbalance, so going into the opposite direction. When we know that we can then study function of these receptors that are coming from the humans, then we started to collaborate more, and we put together a study with all the people in the Mitchell Center, Dr. Calle, Dr. Balaye, Dr. Telatela, Dr. Fracassi, and many other people, to study particular functional aspects of these individuals. And then we use a technique that is called fast LTP, that was developed by Aleph Prieto in California, Irvine. And basically the technique is that you do chemical potentiation in synaptosomes that are isolated from postmortem tissue. And then you can use antibodies that are going to bind to the receptors that have been inserted by the potentiation. And these isolated synaptosomes, you can do flow cytometry, and then you can measure plasticity in every single synaptosome there. So this is a powerful technique because you can use animal models, but also you can use postmortem tissue. or cryoprotected. You don't have cryoprotection, it is a bit of reduction of the efficacy, but you can do it in any way. Then in the bottom panel, you can see the effects of the chemical or synaptic plasticity in isolated synaptosomes. In the hippocampus, in resilient individuals, you have a small reduction, but it's not significantly different from the controls. So you have some preservation of synaptic plasticity in the hippocampus. And in the frontal cortex, the plasticity is well maintained. So this indicates that resilient individuals, despite the pathology, synapses are well protected in the functional aspect, at least of the plasticity. We also measure AMPA receptors and GABA receptors, and what we observed, as you can see there in the panel, the slope that is in the bottom is the slope between Alzheimer's disease, response between AMPA receptors and GABA receptors, and the one in the higher is the control, and NDAN has almost a similar slope that the control. So this indicates that there is an imbalance in the frontal cortex of Alzheimer's disease, but in resilient individuals, the excitation to inhibition balance is well preserved. Because we wanted to go to the most extreme levels of resilience, then we started to collaborate with Dr. Dirk Kinney and Dr. Paul Crane. They are in the University of Washington, and they are working in the aging, dementia, and TBI study that is part of the Allen brain. So their adult changes in thought study they contain close to 5,000 individuals and about 1,000 autopsies. From these 1,000 autopsies, they were able to get about 9, now probably a little bit more, but at the time when we were talking about the collaboration, they were able to get 9 resilient individuals out of these 1,000. So these are people that are old, and they have the most severe category of pathology. These are actually the cohort. So they have individuals that they call resistant, that are very old people, almost getting to the 90s or beyond that. And they do not have pathology. They don't have tau, they don't have neurofibrillary tangles or beta amyloid. So they call resistant to pathology. And you can see in the amyloid plaque distribution that is very, very low, close to 1. And then the match AD, that's Alzheimer's disease. In the right, you have the resilient and the Alzheimer's disease with resilient. Neurofibrillary tangles, you have, again, the resilient and the matched resilient. And all this and the CIRAD, you have the same. Again, the worst levels of pathology, but nevertheless, as you can see in the panels below, they don't have a delay in the cognitive scores. To the right, you have the resilient individuals. It is using CASI. That is a metric that they developed, Paul Crane developed, similar to goes to from 0 to 100. And then you can see how it doesn't go to the threshold of impairment. They match Alzheimer's disease, you can see how they go down. And the resistant is the same, so in the right part of the panel. So this cohort, we were able to get it. And then Adriana Gutierrez-Grevenkova, that is a postdoc in my laboratory, started to work with them and doing what we do in the lab, that is taking the parietal cortex, that is part of the default mode network, very important for early Alzheimer's disease, and part of that regulates the whole function of the brain. And we took the parietal cortex, isolate synaptosomes, send them to proteomics. So we have all the proteins that are in these synaptosomes. We also did RNA sequencing. So we know the bulk RNA, the gene expression of this particular brain region, the proteomics that is in the synapses that we're going to be studying. And then we did the transplantation. So we can correlate the function of the function of the receptors with the proteins that are making up this function. And in these studies, what we were able to show is that there is a lot of proteins that actually maintain the synaptic function. And those proteins, this is particularly in Alzheimer's disease and mild cognitive impairment. So these proteins, basically, the ones that positively correlate with the function of the receptor, with the amplitude of the receptors, are the ones that negatively correlate with pathology. And the more amplitude of the responses you have, the better is the function of the receptors, and the better are cognitive scores. And you have a set of proteins that are the opposite. Set of proteins that when they are increased, you have changes and alterations in the function of the receptors, worse cognition, and increased neuropathology. And the EA imbalance is one of those factors. So by using this methodology, what we started to see in resilient individuals is that inhibitory at the level of gene expression is that inhibitory system seems to be enhanced in resilient individuals. The points that you see in green are, one point is a single individuals, and we have gefferin in the bottom. Gefferin is a protein that anchors GABA receptors at the synapse. You can see in green that there is a slope. The more gefferin someone has, the more GABA receptor amplitude or activity you have. In Alzheimer's disease, the slope is reduced. So that means that you need more gefferin in the synapses to be able to get to the same level of GABA receptors amplitude. However, in resilient individuals, there seems to be an increase of the efficacy of gefferin. You need less in order to get more GABA receptors. So this is very important, at least in our view, because it means that we have some compensatory mechanisms, again, in resilient individuals. And one of the questions is whether in resilient individuals there is some pathological processes going on. And what we observe in the right panel is basically a gene ontology enrichment, where we not only correlate gefferin, but correlate all the proteins that are in our proteomic samples with the function of GABA receptors. And what we observe here is, in Alzheimer's disease, you have a set of proteins that are associated with toxicity and are unique to Alzheimer's disease. Then, in the controls, you have a set of proteins that are enriched with vital processing in the neuron: synaptic signaling, respiration, mitochondrial function, and so on. NDAN, interestingly, have markers for both. It has markers for toxicity, and it has markers for function. But if you see the color, the darker the color, the more enrichment is in that particular area. So all the regions that are common to Alzheimer's disease and to controls are more significant in resilient individuals. We also measured glutamate receptors. And basically, what I'm just going to show here is that there is a difference in how AMPA receptors are being active in resilient individuals compared to the controls. What we have is we apply drugs like kinase that keeps the receptor open for longer period of times, and then we compare how the same receptor responds to kinase, that is a drug that we use, or to the endogenous agonist glutamate. And we take the ratio, and you can see that there is a gradient between red Alzheimer's disease, green controls, and blue resilient individuals. Interestingly, when we do correlational analysis, what we observe here in this circle is basically the length of the arc is the number of proteins that are correlated with this functional data. Alzheimer's disease is in red, and in control you see is a quarter of the circle. Then half of the circle are the number of proteins that are engaged in resilient individuals compared to the others, meaning that we have a larger amount of proteins engaged in the function, and then the purple lines connect, basically, is the same gene in different lists. So NDAN has genes that are unique to Alzheimer's disease and unique to controls. That indicates that we have pathological processes going on. And then on the panel on the right, we basically the same thing that we observe in transcription. Darker in resilient, that is the right, this one actually. This one, this is the resilient. So we have darker colors indicating more engagement and more compensation. We did, AMPA receptors are made by 4 subunits. You can see in controls and Alzheimer's disease and resilient. These 4 subunits are AMPA 1, 2, 3, and 4. The most abundant receptors in the brain are the heteromeric receptors made by the combination of 2 and 3. And you can see that they are correlated when we measure them. In Alzheimer's disease, this correlation is lost look similar to disruption. But in resilient individuals, we have a higher correlation of these receptors. We did those responses, and interestingly, we didn't observe any difference in affinity between the controls and an Alzheimer's disease. The green and the black line over there, they are those responses for AMPA receptors in controls in an Alzheimer's diseases. They are identical, one on top of each other. In the resilient individuals, we just see a little bit of change in the slope, but nothing else. The affinity is maintained. So we also measure AMPA receptors that are permeable to calcium, and we only observe a reduction of AMPA receptors that are permeable to calcium in resilient individuals, suggesting that we have some changes in the subtypes of the receptors, but just to keep the function going on. And one of the most important things is that when we put all this together to do a system biology analysis and see which are the genes that are common across all the pathways in the compensation of glutamate receptors and GABA receptors in this cohort, what we observe, this is very busy, but you have there on top the control, AD, and resilient. I just want to take your attention to the logarithm of the scale below. It's 15 in control, 20, but in resilient is 50. That means, and that line is the cellular respiration. So one of the most important changes that happen in resilient is particularly the enhancement of mitochondrial respiration, indicating again compensation. So then, one of the conclusions is that the cognitive function in resilient individuals seems to depend at least on part of efficient microglia removal of synapses that preserve axonal viability. Then by directly recording the function of the receptors, we can see that there is evidence of pathology, but also strong compensatory mechanisms. Seems to be a tug of war between toxic and compensation that maintains, at the end, at least in resilient individuals, seems to be protecting them from the disease. So promoting, understanding how this is happening and promoting these mechanisms can probably help to bring some therapies for people that are vulnerable. And in the Milchel Center, we are putting together, we are working with this for a while, but we are even putting together more work between the researchers that we have. We recently got Claudia Marino, that was part of the work to identify the Chris-Church mutation and the Colbos mutation that are protective for resilient. We have Dr. Fracassi, Dr. Kayer that is doing, we host the polymorphs network and can provide brain-derived tau oligomers for everyone that wants to have them. And then with all the work that we've been doing in the Milchel Center, and Dr. Tagliatella was able to receive a philanthropic donation of $25 million, where he was able to fund the Moody Brain Health Institute, and has been very, very helpful putting together some of the work that is going to be doing, and where TARCC has been very, very important, because TARCC has provided samples for biomarkers that we are doing. And in this effort, we want to bring together the people like TARCC. You guys, Texas, as was mentioned, is one of the best places to be right now. In the Milchel Center, we like to collaborate. We have several collaborations. If you're interested in also doing or receiving some of the samples that we have, just contact me or contact Dr. Tagliatella, Dr. Kayer, or any of the other researchers, and we are always happy to collaborate. This is just part of the funding that we are receiving, part of my laboratory. And I don't think if there's questions or not questions. There's no questions. Well, just thank you very much for your attention.
Ladies and gentlemen, please welcome back Dr. Rodrigo Morales.
All right, so I'm here to announce the winners for the posters for this year. And we have 4 winners. They are equally ranked. We don't have first, second, third place, no. They are all equally ranked, so I'm not making it's a random order. So in order to get your certificate, please see TARCC stuff. But when I name you, please stand up so everybody can see you. So the first winner here is the title of the poster is Unraveling Synaptic Tauopathy in Alzheimer's Disease, the role of A-beta and selective neuronal vulnerability in tau spreading and synaptic dysfunction. The presenting author is Srinath Kadamungi from UTMB. So congratulations. So the second title is Montreal Cognitive Assessment Memory Scores and Alzheimer's Disease Neuropathological Change. Presenting author Oscar Kronberger from UT Southwestern. Okay. Third poster, Developing Novel Therapeutic Inhibitor Peptides for Alzheimer's Disease and Related Diseases. Presenter author is Heidi Mendoza Oliva from UT Southwestern. And the fourth one is Python-based machine learning for three-dimensional human brain model segmentation and analysis. Presenting author Morgan McCallie-Smith from UT San Antonio. All right, so I want to introduce Dr. Cullum again for some concluding remarks.
All right, we made it, and we're at the end now. Now, for those that have left already and early, you're going to miss out. We're actually going to do a large group photo, I believe. Our photographer is here somewhere. Is our photographer here? And we know where we're going? Okay, we're gonna make a caravan. There's gonna be a large. He's gonna like do an aerial sort of photo of everybody all at once. But I want to thank you all for coming. Stay for the photo and safe travels back. And we hope you've had a great meeting. And we look forward to more collaborations and interactions in the future. And for TARCC, January 22nd next year as well. The announcements will be forthcoming. Thank you all very much.